The Netherlands, home to tulips, windmills, cows, dikes, and a nationwide environmental crisis. The Netherlands suffers from a nitrogen crisis. Too many compounds containing nitrogen atoms are emitted. The Dutch nitrogen surplus is even 4 times higher than the average of the European Union. When these nitrogen-containing compounds deposit in nature, they cause acidification and eutrophication. As a result, plants that thrive on nitrogen outcompete those that do not. Eventually, this leads to the disappearance of animals that depend on these plants to survive. In 2016, 70% of Dutch nature areas exceeded critical limits for nitrogen. Excess nitrogen deposition in nitrogen-sensitive nature areas thus significantly threatens biodiversity and habitat longevity. One of the main nitrogen-containing compounds emitted in excess is ammonia. The greater part of these ammonia emissions originate from agriculture, more specifically from farm animals. In 2019, over 49% of all Dutch ammonia emissions originated from cattle alone. Even though Dutch ammonia emissions have significantly decreased since the 1990s, this decline has stagnated around 2013, and the Netherlands is at risk of not making international emission ceilings. Therefore, a commonly mentioned solution is to curb Dutch agriculture to the extremes of halving livestock numbers. However, despite its small size, the Netherlands is the second largest agriculture exporter in the world. Curbing Dutch animal agriculture is therefore not as easy as it might seem. Not only would it have a significant impact on the income of farmers in this vast industry, it would also threaten worldwide food security. Moreover, these measures have led to a clash between government and farmers, resulting in nationwide farmers' protests. All in all, the Dutch nitrogen crisis is a complex problem with economic, environmental, and agricultural demands opposing each other. But what if we can address these demands at the same time? What if we can create a solution based in synthetic biology that works alongside other existing solutions to reduce the burden both on the environment and on farmers? We are iGEM Groningen 2021, and we present to you BYE-MONIA. BYE-MONIA aims to reduce the ammonia emissions at the source, the digestive tract of cattle. Our solution consists of 3 steps.
We engineered Saccharomyces species to produce Alpha-Amylase, the glycosidase that can serve as a feed additive. Theoretically, once added to the feed, Alpha-Amylase will decrease the ammonia emissions by cows while enhancing their milk production and growth. We chose to engineer Saccharomyces species since they're easily amendable with multiple modular toolkits available and they're safe LM1 organisms. Moreover, they are a non-microbial cell factory and they cannot fixate nitrogen.
Step 2. Besides reducing ammonia emissions, we plan to place a filter in barns that contains a special material called a metal organic framework, or in short, a MOF. MOFs are powders that contain metal ions and organic compounds shaped in an orderly fashion. Because of this structure, MOFs contain cavities of several nanometers, which can trap ammonia that flows through them.
Step 3. In the last step, we will close the cycle. We will bring the fill-up MOF back to the lab, where our GMO is. In the lab, we will heat up the MOF, causing it to release the ammonia that it trapped. Then, the MOF can be reused in the farms, while the ammonia will be given back to our GMO, as the Saccharomyces strains that we are using can feed in ammonia as the only nitrogen source. This way, the circle between the lab and the farms will be closed.
Overall, BYE-MONIA should uphold our 5 key values. It should be safe, circular, target the source, create more products with less resources, and be beneficial to farmers.
So how exactly does Alpha-Amylase reduce ammonia emissions by cows? To understand, we first have to understand how cows produce ammonia. It all starts when urine from the cow mixes with feces. The urine contains urea and the feces contain urease, an enzyme which can break down urea. When urine and feces mix, therefore bringing urea and urease together, urea gets broken down into ammonia and carbon dioxide. To understand this process even better, we should zoom in on the first stomach, the rumen of the cow. When you feed a cow, you're actually feeding the microbes in the rumen, which in turn feed the cow. These microbes ferment carbohydrates and protein from the feed, which produces nutrients for themselves and nutrients for the cow. The microbes first break down carbohydrates into oligosaccharides, carbon dioxide, methane, and volatile fatty acids. Oligosaccharides are broken down even further to monosaccharides, which are used to produce microbial ATP. Both carbon dioxide and methane are belched out by the cow. Lastly, volatile fatty acids enter the bloodstream and travel to the liver, where they are converted to ATP. This ATP makes up 70% of the cow's energy supply and is used for milk production and growth. Microbes also ferment protein in the feed. Proteins are broken down into polypeptides and consecutively into amino acids. The microbes can either take up these amino acids directly or break them down even further to ammonia, which they can also take up. Any ammonia that's produced but does not get taken up by the microbes enters the bloodstream. Once it gets to the liver, ammonia gets converted into urea, which is excreted in the urine. Microbes need both energy from carbohydrate fermentation and nitrogen from protein fermentation to grow and multiply. If either is in short supply, microbial growth is retarded, and so is the fermentation of the feed. When the microbes are less able to break down carbohydrates into oligo and monosaccharides, they produce less ATP. The microbes therefore become less efficient at taking up and using ammonia, but will still produce it. Therefore, more ammonia will go to the liver to be converted into urea, eventually leading to more ammonia emissions. In short, if carbohydrate fermentation is suboptimal compared to protein intake, cows will emit more ammonia. However, theoretically, if we add alpha-amylase, which is a glycosidase, it will break down carbohydrates in oligomers. These oligomers are more easily fermentable for the microbes. The microbes will therefore produce more ATP and thus be able to take up more ammonia, causing less ammonia to be converted into urea, ultimately causing less ammonia emissions originating from cattle. Moreover, it is expected that since carbohydrate fermentation is enhanced, more volatile acids will be produced, therefore enhancing the energy supply of the cow. Therefore, the cow should grow better and produce more milk with the same amount of feed.
We decided to test 4 different alpha-amylase genes that have shown to work at the same pH and temperature that the one found in the cow's rumen. The host organism from which these sequences come from are Aspergillus oryzae, Bacillus amyloliquefaciens, Bacillus licheniformis, and Bacillus subtilis. The alpha-amylase gene has 2 main features: the secretion signal and the chain that encodes for the extracellular active enzyme. When we created our construct, besides using the native secretion signal, we also tried different secretion signals that have proved to work in yeast. Overall, our construct consists of a promoter, the secretion signal, the gene, and the terminator. But it doesn't stop there. In order to explore for the capacity of our device to express active alpha-amylase, we took advantage of the modularity that the Golden Gate assembly technique allows for several combinations. Overall, we tested 10 promoters, 4 secretion signals, 4 genes, and 4 different yeast strains. Taking all of that together, we created a combinatorial library containing 640 possible different combinations.
Testing all 640 combinations in order to find the combination yielding the highest alpha-amylase activity is both inefficient and infeasible. Therefore, we aim to test only a small subset and use machine learning in order to make inferences on which combination is likely to yield a high alpha-amylase activity. The automated recommendation tool, or ART for short, was used for this very purpose. This tool uses a Bayesian ensemble model in order to make predictions on untested combinations of parts.
Within our wet lab and dry lab, we set up 4 proof of concept that we wanted to achieve. The main challenge was to create a GMO that can both grow in ammonia and ensure an optimal alpha-amylase activity. For the first proof of concept, we grew 6 different yeast strains in different ammonia concentrations. From this, we selected the 4 best growers to continue our project with. For the second proof of concept, our aim was to use Golden Gate assembly to successfully clone the construct in Saccharomyces species. During the production of the cassette plasmid, we used Ecolite to amplify the DNA plasmid. And once the cassette plasmid was formed, we transformed it into Saccharomyces species. By growing the final strains in selective media and by using GFP screening, digestion, nanodrop, and sequencing, we were able to say that we managed to successfully clone 63 cassette plasmids.
For a third proof of concept, we aimed to confirm that a GMO showed alpha-amylase activity. To do this, we performed quantitative and qualitative experiments. For quantification of alpha-amylase activity, we used an alpha-amylase assay kit. This kit gives a colorimetric output when the substrate, a starch analog, is broken down, resulting in p-nitrophenol. For this, we made overnight cultures of our samples in triplicates, after which we separated the cells from the supernatant. The supernatant was put in a 96-well plate and the absorbance of the plate was measured at 405 nanometers every 5 minutes. The higher the absorbance, the more substrate was broken down, and thus the higher the alpha-amylase activity. We managed to demonstrate alpha-amylase activity, which differed substantially between our samples. As a secondary confirmation, we also performed a qualitative analysis using a starch breakdown halo assay. In this test, samples were plated on a plate containing starch. If the sample shows alpha-amylase activity, halos will be visible around the sample after the addition of iodine. In this experiment, we used cells, supernatant and lysate product from 9 randomly selected samples. Just the plated cells did not show any halos. However, the plates with supernatant halos were visible around several samples. After we lysed the cells using a bead beater, halos were also present around several samples. This indicates that alpha-amylase is present both intracellularly and extracellularly.
For our fourth proof of concept, we tried to find combinations that will have a high alpha-amylase activity using the automated recommendation tool. We initially set out to test 64 out of a total of 640 possible combinations. As can be seen from the frequency distribution of the parts, there are no parts that were substantially underrepresented in the subset. However, throughout the experimental process, 17 combinations were excluded, which eventually resulted in some parts becoming underrepresented. This was also reflected by the predictions made by the art, as the exploratory recommendations suggested testing combinations that contained one or more of the underrepresented parts. Nonetheless, the art was able to fit the training data relatively well, with an R-squared value of 0.63. This gives an indication that the model is likely to be generalizable to untested combinations. Based on exploitative recommendations, we concluded that there was a clear preference for alpha-amylase genes and secretion signal. Moreover, several promoters outperformed the others. But for the chassis, no clear preference was observed.
To ensure that BYE-MONIA is responsible, safe and good for use in the real world, we laid a major focus on human practices. Throughout our entire project, we focused on background research to understand the scope of our problem, and we filled in several brainstorming tools over several months. In addition, we set up 9 safe by design choices to ensure the safety of our project, and we spoke to multiple stakeholders, even creating a framework so that next iGEM teams can ensure informed consent and data protection in their projects.
In addition, we created and implemented educational materials based on synthetic biology. We created a comic addressed to children called Adventures in the Farm, where a group of microorganisms are starring. In the chapters, the microorganisms are faced with a problem, and synthetic biology helps them to overcome it. Moreover, in order to reach an older audience, we also created a web series called The Science Behind the Comic, which covers the scientific principles behind the comic more in depth. Our materials have been implemented all over Europe, and kids from the Netherlands, Bulgaria, Croatia and Spain have met Musoke and Mark.
The ultimate goal of BYE-MONIA is to keep the GMO in the lab and deliver a feed additive to farmers which is ready to use in cattle feed. Next to that, a moth can be placed in pig and poultry barns to capture ammonia, which will be fed back to our GMO. In order to create a safe product for humans and the environment, 9 different safe by design measures will be implemented, most notably biocontainment in the form of a kill switch,
so that our GMO is not able to survive outside the lab. After the starch breakdown test, it was visible that the alpha-amylase produced in Saccharomyces species is present intra and extracellular. Therefore, if our feed additive will be produced on an industrial scale and we want to increase the yield of alpha-amylase, both the supernatant and intracellular fluids need to be used. In order to achieve this, it will be needed to lysate the cells. By performing cooled grinding with glass beads, large volumes of Saccharomyces species cultures can be lysed without denaturation of alpha-amylase.
After the experimental wet lab phase and the processing of the quantitative data from the alpha-amylase kit, the ART recommended testing several combinations of promoters, sequel sequences and alpha-amylase genes. For further development of BYE-MONIA, these recommended combinations should be tested prior to large-scale production of alpha-amylase to better determine the ideal combination.
More research on alpha-amylase is also needed. As of now, the working of alpha-amylase is only proven in theory, and more practical experiments are needed to prove that alpha-amylase helps reduce ammonia emissions and enhance growth and milk production of cows. Moreover, research is needed on what alpha-amylase does to the microbiome of cows and their methane and carbon dioxide production. Lastly, we ourselves were not able to build and test the MOF in 8 months, and more research is needed on the state-of-the-art material.
Even though BYE-MONIA alone might not be able to solve all the problems created by the Dutch nitrogen crisis, and additional research is still needed, we are proud of all we have accomplished over the last 8 months with 9 people. Not only have we managed to successfully clone alpha-amylase into Saccharomyces species, we have also created a combinatorial library with over 640 combinations. These combinations have been analyzed by a machine learning tool designed for our project. In addition, we have also ensured the safety and responsibility of our project with extensive human practices work. And we have designed and implemented educational material for the young and the old. All in all, we are sure BYE-MONIA can contribute towards reducing the burden on both the environment and farmers by turning ammonia into BYE-MONIA.